Hello guys, I hope you all are doing well. Welcome back to my channel. Today I'm going to talk about microscopy and staining technique. First of all, in order to understand what is a microscopy and what is a staining technique, we first need to understand the basic of it. First of all, microscopy is the science of using microscopes to view objects that cannot be seen with the naked eyes. These objects are often tiny, like cells, bacteria, or even molecules. On the other hand, staining technique in microscopy. So, staining is a technique used in microscopy to color cells or structures to make them easier to see and differentiate under a microscope. Most cells are transparent, so staining gives contrast, which helps scientists or doctors identify specific parts of a cell to detect microorganisms such as bacteria and parasites. Now, starting with the PPT, microscopy and staining technique. Revealing the invisible world, unlocking the secrets of cellular structure and microbial life through advanced visualization method. Why microscopy matters? First of all, guys, microscopy and a microscope is a different thing. So please don't get confused over here. A microscopy is a technique of observing different objects under a microscope. Okay. It is not a microscope. Microscopy is the science of using microscope. It is a technique of observing objects that are too small to be seen with the naked eyes. So microscopy is a technique of using microscope. So microscopy serves as our window in the microscopic world, enabling critical discoveries in medicine, biology, and research. From identifying disease causing pathogens to understanding cellular mechanisms, these techniques are fundamental to modern science. Now, if we talk about different types of microscope, there's light microscope, there is electron microscope, there is fluorescence microscope, there is confocal microscope. But the two most common microscope which we all mainly study in our bachelor's or post graduation is light microscope and electron microscope. Also, studying about fluorescence microscope and confocal microscope will be a beneficial thing. Disease diagnosis and treatment planning, drug development and testing, understanding cellular process, quality control in manufacturing. So this is the importance of this is the importance of the importance of using the technique of microscopy. This is the reason why microscopy is important. The very first reason is disease diagnosis and treatment planning, because microscopy is the science of using microscopes, and staining technique allow us to see microscope by using different kind of stains, because some microorganisms are transparent, so without using a stain on them we can't usually see them. It won't be visible to us. So if these microorganisms won't be visible under a microscope, so we won't be able to diagnose a disease or treatment for a certain type of disease. Then next comes the drug development and testing. Obviously, if a microorganism is identified, so according to that microorganism a drug and different kind of test is developed. Then there is a third one is understanding cellular process. It's a very common thing. If any certain microorganisms is observed under a microscope, so through the observation we can say many things about the cellular process, about their compartments, about their organelles. Then last comes the quality control in manufacturing. Of course, there is a lot of quality control measures taken in the lab, which is very important for manufacturing. Also, many quality control measures are taken in the different kind of industries, pharmaceutical company, which is very important for manufacturing different kind of products. Now, light microscopy fundamentals, basic principle to key components. Light microscopy fundamentals In easier word, I will tell you, a light microscopy uses visible light and lenses, and it's common in biology labs. You can find a light microscopy very commonly in any science department, or any life science department, and it has a magnification up to 1000x. It is commonly and mainly used for viewing cells and tissues, because there are also many biology students. I think this kind of microscopes are sometimes available in school also, which is very much useful for O level and A level students who are a PCB student, which makes them easier to visible tissues and cells under a microscope. So basic principle: uses visible light and glasses lenses to magnify a specimen up to 1000x magnification, as I said. Then there is key components: objective lenses, eyepieces, condensers, and illumination system work together. Then there is a resolution limit: maximum resolution of 0.2 micrometer due to wavelength of visible light. While light microscopy offers excellent visualization of cellular structure, its resolution is fundamentally limited by the physics of light diffraction, making electron microscopy necessary for finer details. So, next comes the electron microscope. Electron microscope use beams of electrons for much higher magnification. Can see very small things like virus, or the structure of the cell plant. So the reason why people also prefer using electron microscope is that, because light microscope do not assist in viewing every kind of cell. Sometimes the scientists or the researchers have the need to view even the tiniest cell, which tiniest cell which they find through their research. So it's not possible to view it under a light microscope. So in order to view those very small things like even viruses or structure of the cell plants, they use electron microscope, because electron microscope uses beam of electrons for much higher magnification. There are two types of electron microscope: one is transmission, and one is scanning. The transmission, which is known as TEM, and the scanning, which is known as SEM. In TEM, which is known as transmission electron microscope, it looks through a thin sample, and SEM, which is scanning electronic microscope, it looks at the surface of the sample. So yes, this is why many researchers or many scientists, they prefer using a electron microscope instead of a light microscope. But also in many common biology labs, in many common life science department, light microscope is also present to view cells and tissues. This is the picture of a light microscope, if you can see. Now, now comes the electron microscope. Oh my God, what happened? So you can see scanning electron microscopes, and how you can view things how things look under an electron microscope, SEM versus STEM. You can see this. This is under a scanning electron microscope, how things look under a scanning electron microscope. Now the sample preparation, the foundation of quality imagining. Now the first one is the fixation. Preserve cellular structure using chemical fixatives like formaldehyde to glutaraldehyde. Then number 2 is dehydration, removes water through graded alcohols. It is to prepare for embedding. Then embedding infiltrate tissue with paraffin or resin for structural support during sectioning. And then sectioning creates thin slices using microtomes for optional light penetration. This is how a sample preparation is done. Then the basic staining principle. As I said, that staining is a technique used in microscopy to color cells or structure to make them easier to see and differentiate under a microscope. So this is a basic staining principle. Dye chemistry fundamentals. Staining techniques rely on the chemical affinity between dyes and specific cellular components. Understanding these interaction is crucial for selecting appropriate stains. So you can't use every type of stain on every type of microorganism. For that you need to understand the chemical affinity between dyes and specific cellular components. Then only you can use a particular stain for a particular microorganism to in order to view it properly under a microscope. Otherwise you can just randomly go into the lab and just grab one stain and just put it on any glass slide and start viewing. It won't be possible unless or until it has a affinity between dyes and the specific cellular components. Then comes the cationic dyes. Positively charged dyes bind to negatively charged structure like nucleic acid. Then there is anionic dyes. Negatively charged dyes bind to positively charged proteins and cytoplasm. Now there is common staining techniques. There is also advanced staining techniques which people use, but right now these are the common staining techniques, which is hematoxylin and eosin, gold standard for histopathology. Hematoxylin stains nucleic blue, purple. Eosin stains cytoplasm and extracellular matrix pink red. Then there is a very famous experiment which we all do in microbiology lab, which is gram staining, in order to identify the gram negative and the gram positive bacteria. I will be also showing that maybe in my next video. It will be coming in the next video or maybe next to next video, but I will be surely showing what is gram staining and what is a gram positive and what is a gram negative bacteria. Right now let's just go with the definition of it to understand its overview. So differentiate between bacteria based on cell wall structure. Gram positive bacteria retain crystal violet purple, crystal violet, and gram negative appear pink. So it's very common that if you want to differentiate the gram between gram negative and gram positive bacteria, the gram positive bacteria retains crystal violet, and it also appears to be violet under a microscope, whereas the gram negative bacteria appears pink under a microscope. Then there is methylene blue. Simple stain for basic cell morphology, excellent for live cell observation and nuclear details in simple preparations. So yeah, methylene blue is commonly used to observe the cell morphology under a microscope, a simple cellular tissue. Then these are specialized staining methods. As I said, this is a specialized staining method, which is known as fluorescent staining. Fluorescent staining uses fluorescent dyes or antibiotics for staining. And also the microscope which is used to observe these dyes are fluorescence microscope. The purpose of using a fluorescent staining method is a target specific protein, DNA, or microorganism with glowing colors. So over here, if you can see, these are specific proteins, DNA. The colorful one on the left side, if you guys can see the colorful ones which is appearing in pink, green and yellow, these are the specific proteins, DNA, microorganism with glowing colors. So these are target specific proteins and DNA. So fluorescent staining technique is specially used to target specific proteins. Sample dye, let's for now we'll not talk about the dye. You just need to understand the basics of it right now. Also, just for your knowledge, the dye which is used, the example dye which is used in fluorescent staining is DAPI, which binds to DNA and glows blue. Then is troubleshooting staining issues. What are the issues which people face while doing the staining procedure? Common artifacts: precipitate formation from old regions, yes, uneven staining from poor mixing, background staining from contamination, over staining marks in cellular detail. Prevention studies: fresh region preparation and storage, proper timing and temperature control, adequate washing between steps, quality control slides for comparisons, optimization tips, systematic approach to troubleshooting, documentation of successful protocols, regular equipments maintenance, staff training and standardization. Now there is a future direction: advanced imagining and digital pathology. What are the future directions of this? AI powered analysis, machine learning algorithms for automated diagnosis and pattern recognition in microscopic images, 3D reconstruction, advanced imagining techniques, eating three-dimensional models from multiple microscope sections. Also, there is digital integration, cloud based platforms enabling remote consultation and collaborative research worldwide. The future of microscopy lies in the integration of traditional technique with cutting edge digital technologies promising unprecedented insights in microscopic world. As I said, that microscopy technique and staining techniques is a very essential thing because it helps in identifying many and various kind of microorganisms, which helps in developing drugs and treatments, and which helps in which helps in identifying different kind of dangerous disease and so on. So it's really important, and it's really one of the most important things. I think it is the first step to discover or develop any drug or the treatment or prevention of any particular disease. If this thing isn't there, so I think maybe the development of different kind of disease or the development of different kind of drug or the identification of different kind of disease won't be really possible. So yes guys, this was all for today. In the next slide, in the next video, I will be explaining about microbial growth and reproduction. Also, I will be in my upcoming videos, I will be showing different kind of staining techniques and how these staining techniques are used, especially the gram negative and gram positive one, especially the staining technique, as I said, gram staining techniques. And there are many videos which is yet to come. So in the next video, I will be talking about microbial growth and reproduction. So that's all for today. Thank you very much. Stay tuned with the next video. Please like and subscribe my channel. Thank you.